Look up at the sky right now. About 78% of the air you're breathing is actually nitrogen gas. It's absolutely essential for life, right? Yet, in its atmospheric state, it's basically a locked vault, totally unreactive. For us, and for most organisms, it's pretty much useless. But plants? Wow, plants have brilliantly solved this problem. So, in this explainer, we're going to trace that incredible journey of nitrogen metabolism. We'll start way up in the vast global atmosphere and zoom all the way down into the microscopic molecular machinery of a single plant cell.
Okay, let's dive into this. Here is our roadmap for today. First, we'll look at the global nitrogen cycle. Then, we'll cover nitrate reduction to nitrite, followed by nitrate reduction to ammonia. After that, we'll unpack the GS-GeoGet assimilation pathway, look at alternate ammonia assimilation, and finally bring it all together with plant amino acid biosynthesis.
Section 1. The global nitrogen cycle, the atmosphere to soil.
You can really think of the nitrogen cycle as this massive global relay race, passing the baton between three main pools: the atmosphere, the soil, and biomass. Step 1 is nitrogen fixation. This is where that stubborn atmospheric N2 gets converted into usable ammonia. And here's a crazy fact for you. Breaking the triple bond of atmospheric nitrogen takes an immense amount of energy. To do this industrially, humans literally need hundreds of degrees of heat and extreme pressure. But these microscopic organisms? They just pull it off flawlessly, right there in the dirt. Step 2 is ammonification. When biomass decays, microorganisms like Bacillus vulgaris step in to convert organic nitrogen back into ammonia and ammonium ions. Then we hit step 3, nitrification. This is a two-part process. First, bacteria like Nitrosomonas oxidize ammonia to nitrite, and then Nitrobacter finishes the job by oxidizing that nitrate into nitrate. Finally, step 4 completes the loop, denitrification. Microbes like Theobacillus denitrificans convert those nitrates back into dinitrogen gas, sending it right back up into the atmosphere. Section 2, nitrate reduction to nitrite, the first assimilation step.
Let's look at this equation to see how it builds the foundation for everything that comes next. This reaction happens right in the cytosol of the plant cell and is catalyzed by an enzyme called nitrate reductase, or NR for short. It's honestly like a beautiful mini-narrative of electron transfer. We start with an electron donor, typically NADH or NADPH if we're looking at non-green tissues, which generously donates 2 electrons. These 2 electrons are then transferred directly to the nitrate molecule, reducing it to form nitrite.
But how exactly do those 2 electrons make that jump? Well, nitrate reductase is made up of 2 identical subunits, and each is equipped with 3 specific prosthetic groups that basically act like stepping stones. First, flavin adenine dinucleotide, or FAD, accepts the 2 electrons. It passes them along to the heme domain, which then hands them right off to the crucial final stop, a molybdenum ion complex to a purine molecule. This molybdenum complex is what actually physically transfers the electrons to the nitrate, and this is absolutely crucial. If a plant has a molybdenum deficiency, this entire relay just breaks down, and that leads to a toxic accumulation of nitrate in the plant.
And this brilliantly illustrates the elegant efficiency of plant biochemistry. The plant actually regulates this enzyme at the post-translational level through reversible phosphorylation. Basically, when there's light and plenty of carbohydrates around, a phosphatase enzyme is stimulated to dephosphorylate a specific serine residue on the NR enzyme's hinge 1 region. That turns it on. But in the dark, a magnesium-stimulated kinase phosphorylates that exact same serine residue. This acts as a docking site for a 14-3-3 inhibitor protein, which renders the enzyme completely inactive. This setup is amazing because it allows the plant to adapt to resource availability in mere minutes, instead of the hours it would take to build or break down the enzyme from scratch.
So we've successfully reduced nitrite to nitrite. But there's a massive catch here. Nitrite is highly reactive, and it is deeply toxic to the plant. So, how does the plant cell safely handle it before it causes catastrophic cellular damage?
Section 3. Nitrite reduction to ammonia. Chloroplast protection.
To protect itself, the plant immediately transports the nitrite inside the chloroplast, where the enzyme nitrite reductase, or NAIR, takes over. Now look at the scale of this reaction. We are no longer dealing with a simple 2-electron transfer. This process requires a massive transfer of 6 electrons to fully reduce that toxic nitrite into ammonium. So where in the world does the plant get that kind of reducing power? It's actually donated by reduced ferredoxin, which gets piped right in from the photosynthetic electron transport chain.
The nitrite reductase machinery itself is pretty fascinating. It consists of a single polypeptide equipped with an iron-sulfur cluster and a specialized heme. The electron flow here moves from the ferredoxin straight through the iron-sulfur cluster to the heme and finally lands on the nitrite. The whole thing is activated by the presence of nitrate and light. But naturally, if the plant builds up a surplus of end products, like the amino acids asparagine and glutamine, that logically signals the enzyme to switch off. It's a perfect way to conserve energy.
Section 4. The GS-GOGAT assimilation pathway, neutralizing ammonia.
Okay, so free ammonium is also toxic, meaning the plant has to instantly weave it into organic molecules. The first step of this rescue mission is handled by glutamine synthasase, or GS. This enzyme takes our newly formed ammonium and combines it with glutamate to forge a molecule of glutamine. Now, this is a highly energy-intensive process. It requires an absolute investment of ATP, and it really relies on the presence of a divalent cation, like magnesium, manganese, or cobalt, to act as a vital cofactor to make the whole thing work.
So the crucial point here is that not all glutamine synthetase is created equal. Plants actually have 3 highly specialized classes of GS, strictly separated by their cellular location and biological purpose. Cytosolic GS is super busy in germinating seeds and vascular bundles managing intracellular nitrogen transport. And interesting enough, it isn't regulated by light. Then you have placidal GS, down in the roots generating amines for local use. Finally, chloroplastic GS is up in the shoots, specifically tasked with reassimilating the ammonium produced during photorespiration.
The second half of this pathway is catalyzed by glutamate synthase, or GOGAT. This enzyme completes the cycle by taking the amide group from the glutamine we just made and transferring it onto a molecule called 2-oxoglutarate. The beautiful mathematical result of this molecular swap? The production of 2 whole molecules of glutamate.
Much like GS, GOGAT comes in 2 specific types, tailored directly to their environment. NADH GOGAT operates in the plastids of roots and non-photosynthetic tissues. It uses NADH as its electron source to assimilate ammonium right from the soil. In stark contrast, FAD GOGAT lives up in the chloroplasts and relies on ferredoxin for its electrons. This contrast perfectly reinforces the overarching theme of how meticulously plants manage their energy sources, based strictly on whether the tissue is photosynthetic or not.
Section 5. Alternate ammonia assimilation methods, backup pathways.
Plants always keep a few backup reversible pathways in their biochemical toolkit. The most fundamental of these is transamination. This is the reversible transfer of an amino group from one amino acid directly onto a keto acid, creating a brand new amino acid. This reaction requires pyridoxal phosphate, which you might know as vitamin B6, as a cofactor. You can really think of this process as the plant's internal distribution network, efficiently shuffling nitrogen away from glutamine and glutamate to synthesize other essential forms wherever they're needed.
Along with transamination, we have enzymes like glutamate dehydrogenase, or GDH, which handles reversible glutamate synthesis. We actually see an NADH-dependent version in the mitochondria and an NAPDH version in the chloroplasts. There's also asparagine synthasase, or AS. This enzyme transfers the amide nitrogen from glutamine onto asparatate to produce asparagine and glutamate. You'll find AS working hard in the cytosol of leaves, roots, and especially in nitrogen-fixing nodules.
Now, what's really interesting about this is how it reveals the plant's real-time decision-making skills. The plant constantly monitors its nitrogen to carbon ratio. When there's high light and plenty of carbohydrates around, it inhibits AS and fires up the GS and FTGOG pathways to favor carbon-rich compounds like glutamine and glutamate. These are used right away to build new plant tissue. But in energy-limited conditions, the plant completely changes gears. It throttles back the GS-GOG pathway and activates AS instead. This smartly favors the production of asparagine, which is super dense in nitrogen, making it absolutely perfect for long-distance transport or long-term nitrogen storage.
Section 6. Plant amino acid biosynthesis, the building blocks of life.
Unlike humans and most animals, who have to forage or hunt to get certain essential amino acids from their diet, plants are entirely self-sufficient. They synthesize all 20 standard amino acids completely from scratch. They do this brilliantly by marrying the nitrogen pathways we've just discussed, pulling the nitrogen from glutamine and glutamate transamination, with foundational carbon skeletons. Those carbon structures? They're pulled right out of core metabolic processes, intermediates like 3-phosphoglycerate and pyruvate from glycolysis, or 2-oxoglutarate and oxaloacetate from the citric acid cycle. Honestly, it's a masterpiece of biochemical engineering.
So, we've traced nitrogen from an inert, unreactive gas in the vast atmosphere, down through the soil, into the plant cell, and all the way into the very building blocks of biological life. It's a heavy, highly intricate system of biochemistry. But I want to leave you with this thought. As our global population continues to grow, how could mastering our understanding of this delicate nitrogen to carbon balancing act revolutionize global agriculture? Could optimizing these exact enzymes be the absolute key to the next green revolution? Thank you so much for exploring this fascinating explainer with me today.